In Chapter 12, we're going to be talking now about the senses and how the senses bring information from receptors, which are all part of the peripheral nervous system, into the central nervous system. And these sensory receptors are critical. And so we'll be talking in this lecture. We'll be talking about sensory receptors and how sensory receptors do this process called adaptation, where they reduce the amount of information that they're bringing in that the central nervous system has to process. So that's our goal, and here we go. So sensory receptors, they are the bits that collect information about the changing conditions that are either inside the body or outside the body. And then their role is to notice these changing conditions and then to relay that information into the central nervous system. And this allows the body to be able to respond appropriately to the stimuli that threaten homeostasis. And so it's all about controlling the homeostatic functions of the body, making sure that things are good. So when we talk about being able to sense something, that's the ability to perceive a stimulus, that changing conditions. So stimuli are those changing conditions. And this concept of a sensation, that occurs when sensory impulses are transmitted to the brain to be interpreted. Perception, then, is that interpretation, the conscious awareness of a sensation and the interpretation of that sensation as we try to make sense of it. Are we bringing in information about changes in the temperature, or instead is it information about sounds that are being made in the external environment? Or a third choice could be, is it about changes in the chemical composition, say maybe the pH of the blood or other body fluids? So perceptions are created in the central nervous system, there, the cerebral cortex, as we interpret a sensation that was delivered to the central nervous system. All right. So when we think about the senses and the ability to detect changes and changing conditions, we can divide the senses into two general groups, or two broad groups. The first are the general or somatic senses. So general senses, these are the senses that have receptors located throughout the body, maybe found in the skin or muscles, joints, and the viscera. And they provide sensitivity to touch, both the light touch that we do with our fingers or that we can feel on other places of our skin. They provide information about deep pressure. They notice conditions that are then interpreted as pain. They keep track of temperature, notice changes in temperature, and in vibration, itch, movement, and then this crazy thing called proprioception. If you took a moment right now, and I'm going to give you a moment to close your eyes, and with your eyes closed, reach out and touch your nose, touch the tip of your nose. And so then open your eyes and kind of notice, did you hit your nose? If you're able to do that reasonably well with your eyes closed, it's because you have a good sense of proprioception. You know, as you bend joints, you know where those bones are now landing. And so you can do something like touching your nose pretty easily. The receptors, the sensory receptors of the general senses, they tend to be relatively simple. They may be a dendrite or a single neuron. Sometimes the dendrite will be wrapped in a couple of connective tissue cells, but still just a very simple organ if there are more than just a neuron involved. The second of the two groups are the special senses. And these are the ones that have receptors located in the head, and those receptors are very complex and associated with sensory organs. And so when we talk about the special senses, we're talking about vision, or hearing, taste, smell, and then last of all, something called equilibrium. Equilibrium is recognition of where your body is in space and what it's doing in space. So we notice acceleration, and we notice turns of movements of the head with equilibrium. That's special sense. Okay, so to really get a good understanding, I was about to say, to get a good sense, to get a good understanding of what, of how the senses work, the first thing we have to understand is sensory receptors. Sensory receptors are the specialized structures that are found at the end of peripheral nerves. Then they are all afferent nerves, nerves that are leading or that are going to be heading toward the central nervous system. And they are capable of detecting stimuli. And stimuli, again, are changes in environmental conditions. And that environment might be the external environment, so that we can notice that, ooh, it's chilly out here, I need to put on a jacket. Or they could be changes in the internal environment. Ooh, it's warm in here, we better start sweating to cool down. So if the sensory receptor is stimulated to a sufficient degree, then that stimulus is translated into an action potential that can then be relayed to the brain along that sensory nerve axon. And then when we get to the brain, when that sensory impulse makes its way to the brain, it undergoes what's referred to as transduction. And that's when we translate a stimulus, a sensation, into I'm sorry, let me go back and try that again. So the process then of, so forget what I just said about transduction. Transduction is instead the process of translating a stimulus, so the change in temperature, into an action potential. And so depending on the kind of information that's being brought into toward the central nervous system, there are going to be different ways that transduction occurs. Okay, so when we think about sensory receptors, we can classify them by the type of stimulus that excites them, that gets them to do this thing of transduction. So one type of sensory receptor is a chemoreceptor. So given that as a prefix, chemo, it's all about, these chemoreceptors are all about noticing and responding to small changes in the concentration of specific chemicals that are dissolved in body fluids. It might be hydrogen ions, keeping track of that. It could be calcium ions. So a number of different chemicals that we can respond to when they're dissolved in body fluids. So those are chemoreceptors. Thermoreceptors, again with that prefix, I bet you can imagine what it's going to be involved with. Thermoreceptors respond to changes in the temperature. Thermoreceptors are generally free nerve endings. So when we use that term free nerve endings, it's free because it doesn't have any cells wrapped around it. It's just a dendrite with no connective tissue around it. And these free nerve endings are found in tissues like the dermis, in skeletal muscle, in the liver, and in the hypothalamus. That's where we see primarily thermoreceptors. Thermoreceptors will produce lots of action potentials when the temperature is changing. So let's say on a summer day, it's hot outside, you're sweating, and you walk into a grocery store and you notice, oh, it's chilly in here. That's because your thermoreceptors are noticing that there's been a temperature change here external to the body. And interestingly, temperature sensations are carried along the same nerve pathways as pain sensations. Who knew? We haven't talked about pain yet, but we'll get there. Pain receptors, these pain receptors, they go by the fancy name of nociceptors. So nociceptors or pain receptors. These are also free nerve endings, and they tend to have pretty large receptive areas. A receptive area is the portion of the body that the sensory receptor monitors. And so if you have relatively large receptive areas around a free nerve ending, that means that there's a lot of places you could stimulate the skin or whatever the tissue is and get it to be excited and have a response. So pain receptors are stimulated by extreme temperatures. So really, really cold or really, really hot causes a sensation of pain, not so much a sensation of either heat or cold. And this is in part why if you've ever been on a sports team and had to do any kind of therapy where you plunge your arm or your leg into a freezing cold ice water bath, it doesn't feel cold, it feels like it's burning. So and so burning pain, that is a pain response. That's a pain perception to the stimulus that we're receiving from the excited pain receptor. So it's stimulated by extreme temperature, by tissue damage, and here I'm talking primarily about tissue damage like twisting or smashing or squishing tissues. So that will also stimulate pain sensation and pain perception. And last of all, chemicals. Bradykinins are one group of these sorts of chemicals. I don't need you to remember that or to memorize that term, bradykinins. But bradykinins are chemicals that are produced by cells that have experienced damage, and in response to bradykinins, then pain receptors they notice those bradykinins in the tissue fluids, and they respond by sending a pain impulse. And pain sensations can be carried on two types of neurons. Type A fibers carry something that is referred to as fast pain. So these are fast pain sensations. They're carried on myelinated fibers. So they have myelin on the axons. Fast pain is also referred to as prickling pain. So this is the kind of pain that you would feel with a paper cut or with an injection from a needle. And the receptive area for fast pain tends to be relatively small, and so it's easier to find the origin. You can point exactly to where you got that paper cut. Whereas type B fibers, they carry what are known as slow pain. So they're carried on unmyelinated axons. So slow pain goes on unmyelinated axons, also talked about as burning or aching pain, and they tend to reach the brain more slowly and are interpreted more slowly then. And with a really wide receptive area, it can be difficult to tell exactly where that burning or aching pain is coming from. So imagine when your back is sore and you get your honey to give you a little back rub, and you're like, oh, yeah, that's it. What? No, that's not quite it. A little to the left. Yeah. Oh, yeah. No, wait, no. Up now. So that is, oh my gosh, how did that happen? That is, that's what slow pain or this burning, aching pain is like. And it comes, it's transmitted along type B fibers. Okay, so we're still thinking about the types of sensory receptors. There are also mechanoreceptors, so with a name like mechanoreceptors. Did I miss one? I think I missed one. Let's go back. Nope, we've talked about pain. We talked about chemo and thermoreceptors. Okay, we're doing good. That's right, I know what happened. I went out of order with your notes because things didn't fit well on slides. So sorry about that. Now we're going to talk about mechanoreceptors. These are receptors that respond to mechanical forces that twist and distort cell membranes. And so rather than being chemically gated, the ion channels that open or close, they close or open when the cell membrane is stretched or twisted or compressed. No, so that T doesn't need to be there. So they open or close if they're twisted, stretched, or compressed. And there are several kinds of mechanoreceptors. Proprioceptors, these are the ones that we talked about that provide that sense of proprioception. They monitor the tension in tendons, at joints, and in the muscles around the joints, providing information about how the body is positioned. Baroreceptors, think barometer here. A barometer measures air pressure. So baroreceptors are about noticing changes in pressure. Where might we see baroreception? Think about in the blood vessels, especially the arteries. We have baroreceptors that notice changes in body blood pressure and respond to that. Last of all are the tactile receptors. A tactile sense is about touching. These detect touch, pressure, and vibration. And they may be just free nerve endings, or they may be enclosed nerve endings with specialized cells wrapped around the cells of a connective tissue wrapped around that nerve ending, or sometimes even they're just like a full-on, just a specialized cell that only is responsible for this touch or sensation. Okay, so that's mechanoreception. And then last of all, when we think about sensory receptors, are photoreceptors. Photo, that means light. And these are specialized neurons that are found in the retina of the eye, and they are stimulated by light. And we see two kinds of photoreceptors in the eye. There are the rods. These are the ones that detect the amount of light that's being produced. So they provide black and white images, and they notice, well, where is it really bright and where is it really dark? The cones are a second kind of photoreceptor, and their job is to notice the wavelength of the light, and thanks to them, we have color vision. So we'll be talking more about photoreception when we get to that special sense of vision. Okay, so we can classify sensory receptors by what they respond to. We can also classify them by their location. Exteroreceptors, the term like X, they detect and they notice changes, the stimuli that take place at the body's surface. So they're busy noticing the external environment. And so they are sensitive to touch, to pressure, to pain, and also to temperature. And we see them in some of the special sense organs. So exteroreceptors in the eyes, also in the ears, in the nose, and there should be a comma there, also in the mouth. So exteroreceptors notice what's going on with the external environment, while interoreceptors, also called visceroreceptors, or visceroceptors, excuse me. So interoreceptors or visceroceptors, these notice stimulus inside the body, either stimulus coming from the viscera or stimulus about or regarding changes in the blood. Generally, interoreception is not perceived by the conscious mind. So the stimulus, the sensory impulse, is transported along neurons in the spinal cord, and it may, and then on up into the brain, brainstem, and the brain. But we don't notice them. We don't take that that sensation and turn it into a perception. So interoreceptors, they monitor primarily changes in pH, in body temperature, in the concentrations of carbon dioxide and oxygen, the stretch that's occurring in a tissue, things like that. And last of all, then, in this classification by location, then we can talk about proprioceptors. They also respond, like the interoreceptors, to internal stimuli, but they're found only in skeletal muscle, in joints and tendons and ligaments, and other types of connective tissue like those. And their job is, again, and we've talked about this already now, they detect body movement and body position. Okay, a third way that we can classify sensory receptors. We can classify them by their activity. Tonic receptors are always active. So they are always checking to see, is there a stimulus coming? Phasic receptors, on the other hand, are usually inactive. But when conditions change, they become active for a very short time. So pain reception is always active. We're always checking, we're sending impulses regarding pain. Whereas these phasic receptors, they only send action potentials during time of change in the stimulus. So, last of all, then, is a discussion about how much information that the central nervous system has to deal with. I've been told that if we actually were we're only consciously aware of about 1% of all the stimulation that is being detected by our bodies. And so that is a very adaptive thing, or can be a very adaptive thing. I suspect that if we were aware of all that information, that it would be, it would be crazy making, because it's just more than we are really able to manage. So sensory adaptation is an important characteristic in sensory reception. So what is adaptation? Well, it's a reduction in sensitivity in the presence of a constant stimulus. So if, let's think for instance now, you walk into your house and you smell very clearly a gas smell, and you're like, uh-oh, I've got a leak somewhere, I need to check this out. So you go sniffing around, and after a little bit, it's hard to smell, and so you have to actually consciously tell yourself, I want to be able to smell this. That's because sensory adaptation has occurred. You've stopped being able to notice that smell. The sensory adaptation will help you if you are in the commons area getting some lunch and you're also trying to study at the same time, because there are 50, maybe 80 people all milling about in the commons area making big noises, and if you can tune them out, then you are doing sensory adaptation. So this sensory adaptation, very it's a very valuable process, because it helps, again, to limit the amount of information coming into the central nervous system. And we can do sensory adaptation in two ways. There's what's called peripheral adaptation, and then there is central adaptation. So let's take a look at those. We'll start first with peripheral adaptation. So if a sensory receptor begins to receive constant information, then that sensory receptor will trigger action potentials at a slower and slower rate if it's peripheral adaptation. So the sensory receptor actually becomes less sensitive to the environment. And so as it slows down the rate of transmission of sensory impulses to the central nervous system will stop paying attention to it, or not stop getting information about what's going on. And this is a characteristic of phasic receptors, those receptors that shut down. And so phasic receptors, they tend to respond very quickly to changes in stimulus level. And as the stimulus keeps coming, then the receptors stop producing action potentials, and therefore we stop getting information about this. So if you were to, imagine it's the first day of summer, school's been out for a couple of weeks already, and you have been waiting because you were really keen to go down to Lake James, let's say, and take a little swim. And so here it is, first day of summer. You're standing on the pier, and you can see that beautiful sparkling water, and you run down the pier, and then jump in. Woohoo! What do you think you feel? It is super cold. In fact, it's so cold you almost come squirting right back up out of that water. But, so when you do come to the surface, you're screaming, woohoo, and yowza, wow, that was crazy. But within about 30 seconds, you're like, oh, okay, I can stand being in this water. And that's because the thermoreceptors have adapted, and in response to that adaptation, we're not getting as much information about how cold that water might be. This could be valuable because, if we were constantly noticing what the temperature of the room was, that'd be a real pain in the rear end. Thermoreceptors, they stop responding when the temperature is stable. So this is not characteristic of those tonic receptors. Those are receptors that tend to respond very slowly to stimulus, and they continue to produce action potentials even after that stimulus that it was responding to goes away. So pain receptors are like this. When you cut yourself, okay, you kind of get it all kind of put back together. You got your, you've done your best now to bandage yourself up, but pain reception continues, keeps going and going and going. Okay, so tonic receptors keep sending information. Phasic receptors slow down. They come out of phase. They stop doing that impulse transmission, and that allows for peripheral adaptation. And it's peripheral because it's the sensory receptor out there at the periphery of the body that is slowing down, that's changing. It's very different with central adaptation. With central adaptation, as our sensory receptors continue to receive continuous stimulation, then nuclei, those are clusters of neuron cell bodies, along those sensory pathways that impulse is traveling, can be inhibited. So these nuclei are inhibited, and the sensory impulses are stopped right there at that nucleus, so that no more information gets transmitted from that particular receptor. No information gets to come in to the brain. So again, it's all about reducing the amount of sensory information that arrives at the brain, and we do it by blocking the pathway. So for instance, signals from olfactory receptors, they are subject to central adaptation. And so you walk into the house and like, Mom made apple pie. Yum, I love her apple pie. But within 30 seconds, you're not smelling apple pie at all. It's also why if you have a baby and you're changing baby's diaper, when you first open that diaper, it's terrible. But by the time you're done, you're like, it's this baby diaper. I don't smell anything. So those sensory receptors will continue to produce action potentials as we're sniffing and smelling. But along the way, nuclei suppress those action potentials from being received. And so with conscious effort, we can unblock and receive those action potentials. But, so we're capable of altering this inhibition that the central nervous nuclei do. Okay, so there we are. We have seen just about everything there is to see in this lecture. So just to quickly review, we've talked about sensory receptors and how we classify them, whether we're classifying them based on the type of signal that they receive, based on where they're found in the body, and based on how active they are, whether they shut down and stop sending signals when environmental conditions are stable, or not changing, or whether they continue to produce action potentials, even if you're not seeing any change in the stimulus that's being sent to the body or sent to the central nervous system. And then last of all, we talked about adaptation, the ability for the brain to not have to deal with a lot of sensory information coming in by severely limiting the amount of sensory information you can bring in. And so we do that both through peripheral adaptation, where the sensory receptors shut down, or through central adaptation, where sensory receptors keep sending signals, but we block it somewhere along that sensory pathway. All right, so there we are. See you in the second lecture.